Welcome back, dear students. In the previous lecture, we already discussed the definition of the bioluminescence, then the phyletic distribution, types, and structure of the bioluminescent organ. In today's lecture, we are going to study the biochemical and the molecular mechanism. Okay, so bioluminescence is a light produced by the chemical reaction within a living organism. Bioluminescence is a type of chemiluminescence, which is simply the term for a chemical reaction where the light is produced. Bioluminescence is the chemiluminescence that takes place inside a living organism. Bioluminescence is a cold light. Cold light means less than 20% of the light generates thermal radiation or the heat. Most bioluminescent organs are found in the ocean. These bioluminescent marine species include fish, bacteria, jellies. Some bioluminescent organisms, including fireflies or fungi, are found on land. There are almost no bioluminescent organism native to the freshwater habitat. Okay, so the chemistry or biochemical mechanism of the bioluminescence. The chemical reaction that result in bioluminescence requires two unique chemicals. First is a luciferin and either is a luciferase or photoprotein. Luciferin is a compound that actually produces light in a chemical reaction. Luciferin is called as a substrate. The bioluminescent color, which is yellow in fireflies, greenish in fish, is a result of the arrangement of the luciferin molecules. Some bioluminescent organism produces synthesize luciferin or their own. Dinoflagellates, for instance, bioluminescence in a bluish green color. Bioluminescence dinoflagellate are the type of the plankton, tiny marine organism that can sometimes cause the surface of the ocean to the sparkle at night. Some bioluminescent organisms do not synthesize luciferin. Instead, they absorb it through other organism, either as a food or in a symbiotic relationship. Some species of the midshipman fish, for instance, obtain luciferin through the sea shrimp they consume. Many marine animals, such as the squid, house bioluminescent bacteria in their organs. The bacteria or the squid have a symbiotic relationship. Luciferase is an enzyme. An enzyme is a chemical which is called as a catalyst that interacts with the substrate to affect the rate of the chemical reaction. The interaction of the luciferase with the oxidase, that is oxygen added. Okay, oxidase is a oxygen added. Then luciferase creates a byproduct called as a oxyluciferin. Most importantly, the chemical reaction creates the light. Bioluminescence dinoflagellates produces the light using a luciferin luciferase reaction. The luciferase found in the dinoflagellate is related to the green chemical chlorophyll found in the plants. Bioluminescence dinoflagellates ecosystem are rare, mostly forming in an warm water lagoons with a narrow opening to the open sea. Bioluminescence dinoflagellates gather in these lagoons or bay, and the narrow opening prevents them from the escaping. The whole lagoon can be illuminated at night. Biologists identify a new bioluminescent dinoflagellate ecosystem in the Humacos Natural Reservoir. Okay, so most bioluminescence reaction involves luciferin and the luciferase. Some reactions, however, do not involve an enzyme, that is a luciferase. This reaction involves a chemical called as a photoprotein. Photoprotein combine with the luciferase and oxygen but need another agent, often an ion of the element calcium, to produce the light. Photoproteins were only recently identified, and biologists and the chemists are still studying their unusual chemical properties. So, photoproteins were first studied in the bioluminescence crystal jellies found off the west coast of the North America. The photoprotein is a in crystal jellies is called green fluorescence protein or GFP. Bioluminescence is not the same thing as a fluorescence. However, fluorescence does not involve a chemical reaction. In the fluorescence, a stimulating light is absorbed and re-emitted. The fluorescing light is only visible in the presence of the stimulating light. The ink used in the highlighter pens is a fluorescence. Phosphorus is similar to the fluorescence, except the fluorescent light is able to re-emit light for much longer period of the time. Glow in the dark stickers are the phosphofluorescence. Then the next is a physical nature of the bioluminescent lights. The first one is a color. The light is blue in some bacteria, Polychaeta and the teleos. It is red in some beetles. Mostly the luminescent light is blue, blue-green or white. Green and yellow are less common. Orange and red are generally rare. In some animals, two different colors are produced by the different luminescent organ. For example, in the pyrosoma, the light is ordinarily bluish in color, but when the temperature is increased, the color changes to red. Second physical nature is intensity of the light. The intensity of light is very low. The third one is a visual efficiency. The bioluminescent light is not at all physically different from other lights. The usual efficiency is high compared with the human eye. So this light can affect the photographic plate and can induce chemical reaction. The fourth is a heat liberation. Bioluminescent light is described as a cold light because the heat produced by the light is less than 0.001 degree Celsius. Then the last one is a wavelength. The luminous light is devoid of neither infrared nor ultraviolet rays. The wavelength of the luminescent light is within the visible range. Okay, so here we stop.